My name is Rob Shibuya. I'm a medical officer in the Division of Anesthesiology, Addiction Medicine, and Pain Medicine. I work in the Center for Drug Evaluation and Research, abbreviated CDER, and we regulate small molecule drugs and biological therapeutics. As I alluded to before, FDA divides regulated products into broad domains, such as human drugs, medical devices, biologics such as cellular and gene therapies, food, veterinary drugs, and tobacco. My organization, DAP, regulates human drugs and therapeutic proteins, including monoclonal antibodies. We cover three areas: anesthesiology, medications for the treatment of addiction, and analgesics. We regulate all analgesics except those indicated for certain specific painful conditions, for example, migraine, endometriosis, and interstitial cystitis. As such, we regulate drugs for the pain of OA. I need to make an important distinction. Dr. Nikolov's division, the Division of Rheumatology and Transplant Medicine, regulates the evolving group of disease-modifying osteoarthritis drugs, also known as DMODs, as everybody in the meeting today knows. DAP only regulates drugs to treat the pain of OA. This is a list of the general types of products currently under development for the pain of OA in DAP. It is not all inclusive. Industry is developing molecules with established efficacy, such as NSAIDs, but is also working on other compounds and combinations, including glucosamine and glucosamine analogs, anti-nerve growth factor agents, other nutritional supplements, and truly novel agents. The routes of administration that we see include systemic, intra-articular, and topical. As I stated before, pertinent to this workshop, DAP's area of authority is drugs and therapeutic biologics to ameliorate the pain of OA. Historically, for many years, DAP or its predecessor accepted an indication that included the concept of the signs and symptoms of osteoarthritis. At the same time, DAP also entertained an indication of just the pain of OA. Evidence to support these indications varied in that the signs and symptoms indication required a win on three co-primaries: the WOMAC pain, the WOMAC function, and a patient global, whereas the pain indication just required a difference in pain intensity. However, with the advent of drugs that might actually affect the signs of OA, DAP has reconsidered this approach. The drugs under development in DAP are not expected to affect signs of OA. Thus, our current paradigm is to require a single assessment related to pain intensity. A clinically meaningful and statistically significant difference in pain intensity will support an indication containing the operative words of the pain of OA. Certain products may contain language in the indication that is related to specific joints to be treated. To paint in broad strokes, in DAP, most OA trials are conducted in patients with OA of the knee or hip. This slide shows typical key selection criteria for those studies. Patients must have documented OA, adequate pain to justify the risk of the investigational drug, and other causes of pain in the index joint must be excluded. I've stressed that for OA products, DAP focuses on the chief complaint of pain. Therefore, the primary outcome measure for studies supporting an indication of the pain of OA are based on an outcome measure related to pain intensity. The generic acceptable method by which patients can report pain intensity is a numerical pain rating scale. The corresponding accepted outcome measure for OA of the hip and knee is the WOMAC pain subscale. Sponsors include a variety of secondary and exploratory efficacy outcome measures for OA studies, and we allow considerable latitude on the secondary endpoints. I have bolded WOMAC function in the patient global because we consider those to be the most clinically relevant efficacy outcome measures for relief of the pain of OA. With regard to the primary efficacy variable, the basic premise is that the investigational product must show a clinically meaningful and statistically significant difference in average pain at baseline compared to the end of treatment. Registrational trials in OA are generally a minimum of 12 weeks of double-blind therapy, although that period may be longer under certain circumstances. Thank you for your attention.